much for spending some time with us this evening. We've got a substantial audience here. I'd say a pretty broad audience, a sprinkle of scientists I recognize. Some of you may recognize. I know you can't see you're not set up so you can see the audience, but I'll do my best. We're in a rather beautiful setting, beautiful church on a fairly warm evening, and very eager to ask some questions.
Well, I look forward to it and thank the audience for their attention.
If I may, I suspect some of the audience are interested in placing what you've done, your accomplishments in the context of science as it is now, as it is today, and in the future. But if I may, I'd like to go back a bit and ask a question from a personal perspective. Two of my best friends in science and two of the best scientists I know also happen to have grown up in a farm like yourself. And I was wondering, is there some secret magic? Is a farm childhood an especially good place for a young scientist to grow up?
Well, I don't know. It worked for me. But I think if you live on a farm, you learn the discipline of work and, focusing yourself and completing a task. And the other thing that is very common is problem solving. You frequently find yourself with something that needs to be repaired, something that needs to be moved and worked. And if you can solve that problem, you can get the job done, or you can refer to an expert. But all those require decisions and getting things done. And I think that type of experience prepares one for sort of being a problem solver in science.
It's been my experience too. At least the importance of troubleshooting. So one more question, if I may, from a scientist, but perhaps you could say the central dogma of molecular biology has been worked out. Does that mean that there's a golden age of discovery that's now over? Are there any big challenges still out there for predicting about entering science and biology? What might they be?
Oh no. Maybe there's not too many secrets hidden within the central dogma itself, but there are, and you can see hints of them in terms of new ideas about RNA methylation and transposable elements. This is all very technical, and then RNA on the surface of cells. RNA has many additional roles that we really do not fully understand. But the more important thing is that there are whole sectors of biological systems that the most fundamental science has to be developed for. These are things that range from the dynamics of systems. We describe systems as equilibrium systems, but they're not equilibrium. They're dynamic. They change. They run on a flow of energy. And we really don't have the conceptual tools to actually deal with the complexity of constructing and understanding dynamic systems. And then we're at that wonderful interface when we're seeing machine learning and AI coming into the field with large data that we've never been able to collect or deal with before. And that's going to take problems that we have been studying on a single cell and turn it into an understanding of how an organ functions, how an organism carries out its regulatory demands during development, and how those things go awry in disease. So, and there are tools in probing these systems with this computational power is going to be radically faster. So no, fundamental science will be essential for maybe forever, but certainly for the next decades. And it's going to be exciting. It's really going to push us to think differently and in different ways.
Thanks. I'd like to turn it back to the audience if anyone would like to have ask a question.
You'll probably have to repeat the question.
Well, let's see if I can speak up. So I was a scientist at CETUS in Chiron back in the 90s, and I still remember interferon beta basically coming to fruition. I remember discussions of RNA being used for delivery. And the question back then was, how do you deliver RNA? And obviously with Alnylam, that basically that delivery mechanism has been successful for vaccines. But from the perspective of drug delivery, when you think of the foundation of companies like Alza founded purely on the concept of how to deliver drugs. The question is, I know that Moderna was focused first on cancer. So in terms of delivery of RNA for cancer treatments, is Moderna still proceeding in that research? Or is it now largely focused on using RNA in vaccine development where the quantities are much less, you don't have to deliver through a whole system, you are basically using the immune system to do the work? So I'm sorry if that's a long-winded question from an actual scientist, not the way, but I'm just curious whether Moderna is also working on the cancer aspects of RNA delivery and where they are with that.
You are correct. And about the first entree in cancer with RNA as a therapeutic tool has been developing and testing, not yet approval, vaccines to actually activate the immune system to different cancers. And there's a very exciting early study out of Sloan Kettering that suggests that pancreatic cancer has, in a small set of patients, has been responsive to an RNA vaccine in which it's been created for that patient with the variants that they identify in the tumor cell. The moving beyond that, I think there's a possibility you're going to be able to use RNA in directing the immune system to attack cells in CAR T cells. But again, that I think is delivering RNA to a small number of cells and seeing the immune system carry out the killing of cells through the body. To try to deliver RNA to every cell, tumor cell in a body, I think is going to be challenging. We're developing technology to direct RNA to different cells, organs in the body now. And we, even in the nervous system, we can deliver enough to modulate genes, and there are clinical trials going on in Alzheimer's and others. So, but I'm not sure we'll be able to take RNA and deliver it into every tumor cell to kill a tumor or change its character. That's going to be demanding, but one never knows what the future holds. But there's lots to be done before we run into any limitation that's going to be significant. Just the whole RNA vaccine working with the immune system would be a godsend for cancer patients, particularly solid tumors.
Any other questions from the audience?
Wondering with the current political climate, namely the defunding of federal defunding of research, if that's going to affect the future of this particular research?
So this is about federal funding of research.
Yeah, yes, whether the federal cuts are going to
it,
affect it.
it, we're apparently in a, well, any reduction in federal funding of the order of 30–50% would be a catastrophe for the country and for science in general and around the world, because we're one of the strongest scientific communities in biological research and biotech and this whole field. I'm hoping that doesn't happen. And when you watch what's going on in Washington, because MIT has interest in that, there's a possibility that what will happen is an agreement on the budget that would put the congressional recommendation in biomedical science at approximately flat funding for a year. That would be 3% relative to inflation decrease, but and it would set things back. And if it continued over years, it would really set back biomedical science. The big challenge for people in the lay is that those reductions will impact how many students are trained, how many people come into the field, how science progresses over decades, and it takes decades to see the impact of really novel, important development and technology. So I'm hoping we won't see a significant cut, anything radical, but I do think it's something everyone should be concerned about.
Yeah, I've got a follow-up to that, and then go back to the audience. But some defenders of those cuts say that basic research could be left to private research institutions instead of publicly funded places like the NIH or publicly funded universities. They could stick to education and let industry take care of the discoveries. You've been really successful in all three environments. What do you think about that?
Well, it's a fallacy. It just isn't possible. The basic research, particularly in biological systems, typically do not generate revenue until they are reduced to practice and proven to be effective. In medicine, it's clinical trials. In agriculture, it's testing in fields and development of markets. And really, it's almost impossible for private sector to fund that type of research. It used to be you could get a monopoly in a particular field like Bell Labs. And Bell Labs then generated the resources to have a very applied but very sophisticated fundamental research on semiconductors and other things. But that in this time of more rapid technology change, the monopoly went away and little bells appeared and they had to be competitive. And now you see things like quantum computing being developed, at MIT and elsewhere, working with companies, but with also a partnership with federal money. So research universities have become really critically important in our whole technology of, producing things that people need. And fundamental research, really, almost all of it is being done in these universities because companies just can't wait 15 and 20 years to see revenues. They can't put their top managers on it. They really have to, be trim and focused to be able to be profitable and supported. So no, industry is not going to step in. And And the seed corn will disappear if it isn't funded by the federal agencies such as NIH and NSF, and then some in DOE and others too.
Thanks. But back to the audience.
It's absolutely incredible to have the opportunity to speak to you. And I thank you very much on behalf of everyone here that you've given us today. I have a question. Do you believe to what extent will RNA be helpful in discovering the miracle for Alzheimer's? And who is doing the best work in that area to see a miracle very soon?
So you might want
Would
to
you
repeat that.
The question was, to what extent will RNA be a driver in finding a cure for Alzheimer's?
Well, we shall see. We know a lot about the disease process and the amyloid process that develops there. And there's an RNAi clinical trial. Alnylam is carrying out that now, where they are introducing RNA into the spinal column and silencing the gene that encodes the protein that becomes amyloid in the brain. And if they can be silenced from the gene level at the messenger level, that might delay and even reverse, but I suspect delay, because once neurons are killed, they don't regenerate in the brain. The progression of Alzheimer's. That's the way to describe it. The one really exciting thing about what we learned in that whole technology of testing and preparing these small RNAs, and this is Alnylam's work, and I was on the board, and I am invested in Alnylam. I have every conflict of interest you can imagine. But the one thing that we discovered is that these RNAs, once introduced into the nervous system, have a very long half-life. And it's possible in patients we could treat once every 6 months or even once a year and maintain the silencing of a gene that might be required for Alzheimer's to proceed. So I would say we're optimistic because the preclinical trials we've done have been interesting and promising, but clearly we will have to learn from the outcome of these patients, and that will take a couple of years. I don't know when it'll first be announced what the outcome is, but I think there's an opportunity there. I'm very excited about it because the treatment of conditions in the central nervous system is one of the biggest unmet needs we have, spanning all the way from Alzheimer's and Parkinson's to many others, Huntington's and ALS, to even schizophrenic and manic depression. So there is real need there. And hopefully some of this technology and maybe other technologies that are being developed will be able to help patients.
Another question from the audience.
I would like your thoughts on the development of guidelines for bio research, what you've experienced and what you might think about the guidelines that should be developed for AI.
Oh, I have friends at MIT
I can repeat.
I think it was about the guidelines um, that should be developed about the use of AI. I have been, first I have to just give you disclaimers. I am not an expert in AI. I have friends at MIT who I'm helping and working with who are trying to apply AI technology to understand biological problems. But in a greater sense, uh, I think I look at the problem in the way that how can AI manipulate people? And I think people have to have access to information, but I would think that information needs to be labeled and taken responsibility for by whoever presents that information. And that is, that's free speech. You just have to declare this is what I'm responsible for because I'm using AI to generate it, and I'm allowing it to be spread. So I don't I'm not wise enough to know how to put an editing machine in society between source and consumer, but I do think we have to be responsible for what we support, and labeling things in a transparent way is pretty standard.
I think there's a follow-up.
With the results of the process of developing the guidelines of the biotechnology that you have been involved in.
Yes. There, yes, there it was very interesting. As you saw in the film, there was uncertainty. Different people had different opinions of the risk. Different people had different motivations for talking about the risk. And some of those motivations were political because it moved people. Fear moves people. Some of it was, certainly legitimate, and some people are just never excited about what the future, if it's going to be changed. And the films shows something that I think is important for us, particularly academics, to think about. And that is, we met with the citizens of Cambridge on the street, essentially, and physically, we met on the street at every street fair in Cambridge that summer, basically setting up a booth, talking about recombinant DNA, showing them that it's people who are doing it, people who live among them, and that it has promised to really improve the quality of their life. So I think there's an opportunity to, in this day and age where there's lots of skepticism about the whole academic world, and which is called the elite world. It isn't elite. It's just a bunch of people doing what they can educate people and try to probe the future and create it. But actually being out and talking about it, showing people who are doing it, answering questions, I think is a very important tool for gaining people's respect. And I think that process is ongoing in AI. I'm reading books about it now, and I think it's a healthy one for society.
How did you feel about the threat to your life at one point, before the Unabomber was called.
What, the breath of my life?
The question was, how did you feel about the threat to your life?
Well, it shocked me. I'm one who tries to understand the nature of the risk and to put it in perspective to the other risks that I am doing every day. Everything is involving some element of risk. Given the mode in which the Unabomber worked, the question was, I needed to protect myself from opening an envelope or a package that could be explosive. And more importantly, I had to protect my secretary and the people in my lab and my family. And so we put, the FBI didn't open all my mail, but clearly screened all the mail that came to me for a month. And I felt actually highly protected by that activity. And I talked to my assistant and everyone in the lab about this issue and making sure that if anything was unusual, they raised it to the attention of others. So, and then the question was I going to move around? And I had a trip planned to California at that time to give a talk at Davis and other places. And I decided that I was going to do that. I just wasn't going to let this control me in what I thought were important things to do. And that's the way I dealt with it. Was it good? No. And I didn't want any publicity about it. And I just went on about my day, and my wife and family did as well. We had some conversations about it, but it was a relief to learn that he no longer would be able to do that.
What we have another audience question from, I believe, our host.
I understand the risks that are out there with AI. I wonder whether you think there are still any risks with the RNA work that's being done, or the RNA work that we see in the future. It doesn't seem to me that there are risks, but then I didn't really understand the Cambridge Frankenstein fear either. So are there, do you see any risks, significant health risks or public health risks in your future RNA research?
It ranges. RNA is involved in a lot of things, all right? COVID is an RNA virus. The viral genome of COVID is RNA and can be manipulated by the tools we've learned how to manipulate RNA. Typically, now we manipulate RNA by making DNA and then making the RNA from the DNA. And that's the way the vaccines are made. So, you can dream about. I don't think it would be easy to do. Well, actually, you should note that the first person who took and made a polio RNA that was infective from DNA was Eckhart Wimmer, who did it at Stony Brook. And it was an experiment that's well known. So there are things that I wouldn't prefer people to do. And there are, everything I do of a nature of an infectious agent at MIT, I have to get approval for doing that genetic engineering type experiment. I haven't done it, though. Let's say I have no ongoing experiments of that nature. But there are things that just don't seem wise and should be done under a high security or reasonable security. And that's true of a lot of biological research. I wouldn't want to see it outlawed or prevented, because the knowledge we get from that type of work is critical for us to understand how to combat pandemic viruses and develop new treatments and new ways of controlling. So if we can't do those types of experiments under the appropriate conditions, we're not going to be prepared when we have something like the last pandemic, and we weren't very well prepared for it. And the RNA vaccine really had enormous impact in protecting people and, in opening activities again. So by and large, historically, good scientific research has been enormously important in creating the world we live in and the standard of living we live in, and how we can protect each other from and treat each other in diseases. So I think we have to proceed with adequate caution and optimism in that the outcome is going to be important for continuing.
Can you tell us a little bit about how the documentary came to be and what the origin story is for how and who inspired you to make this documentary? How did it come about?
The documentary came about because I met Bill Haney, who is the producer and director of Uncommon Production, which produced this documentary. This is his 19th documentary. And he is a friend of a professor at MIT, Tyler Jacks. And Tyler and I work on the same floor across from each other. We published a few papers together over the last 30 years. And I was interacting with Tyler and interacting with Bill in a company they were organizing called Skyhawk, and I'm a consultant to it. And Bill asked me, he had just made a couple of years ago a very exciting and movie or documentary about Jim Allison. Jim Allison is the scientist who developed the immunotherapy to treat cancer. And this has been revolutionary in the treatment of cancer. There were, in many solid tumors, we could, control progression for 2-5 years, but most people continued to progress. With immunotherapy in some of these, particularly in patients with histories of smoking, sunburn and mutagenesis from sun, and DNA repair mutants, you can get cures. Patients never see that cancer again. So, and he's the first person ever to get a Nobel Prize for treatment of cancer. So that documentary I greatly admired, and I know Jim well. And when Bill approached me about this. We talked for a while, and I thought maybe the optimism of this story, uh, which I consider very optimistic, um, the history of, small town boy does well, and the importance of the whole biotech, and particularly as well, the RNA vaccine field, would be a good story to tell now. And I think it's turned out that those are the sort of things that interest people. So I decided that maybe it would be useful, and it certainly would be informative to me to be involved, because I haven't been involved in something like that at that scale. So I decided to participate. My wife, my family, my friends all supported it. I enjoyed the learning and enjoyed somewhat. It's a little like undressing in public. That's never comfortable, at least for me. I'm a pretty reserved fellow. But that's the motivation for doing it.
We have one last audience question. Go.
There was a lot of work done before the COVID vaccine, that decades of research that led up to being able to create the COVID vaccine. Having done that, does that lay out a template so that as future viruses arise, we can be hopeful that we would be able to respond quickly in vaccine development for future potential surprising viruses?
I'm sorry, I'm going to need that question repeated.
Does the rapid development of the COVID vaccine, is that a template on which we could develop future vaccines for future surprising viruses?
Oh, yes, no question. It's already being used to create vaccines for flu and other viruses, RSV. And it is a technology that is very programmable, and it's very dependable in terms of input and output. So as it was for COVID, there were, I think, 30,000 people in a clinical trial showing it worked. Also, in that 30,000 people in that clinical trial, they followed all complications, and the complications were rare. And now it's been used in 1 billion people, and it's clearly effective. There's probably some complications in part, but almost everything has some complications. So it's a wonderful technology. It's one to be celebrated, and one we may need critically again in the future. The whole COVID, and as well the HIV pandemic or endemic, these are viruses that move around as people move around the world, and we're in a new world, and the speed in which people move and how fast the virus can spread. And we're going to need this technology, as well as its implications for treatment of cancer and probably other other treatments that depend on modulation of the immune system.
Thanks. And I'll allow myself a final wrap-up question, if I may. Referencing the film title, Cracking the Code, aside from the genetic code or codes, were there any others, like professional codes, social codes, that you found difficult to crack, and was it hard?
Well, yes. Everyone, I assume, that comes from a community, a small community, that doesn't have a lot of professionals in their lives as extended family members and neighbors, you have to learn how to, working with mentors who know more than you, how to be disciplined in preparing for, discussions with them, how to be precise in your speaking, how to do your homework and try to learn what they're telling you so that you're not wasting their time. And people want to see people work as teams, and you have to learn how to share credit and work with teams. So you just have to be respectful, try to learn and be effective in the outcome. Was it awkward? Sure, sometimes. I was in social settings that I hadn't experienced before, but people support you. And, they're not there to run you down. They're there to basically, help you. And you just depend on their help and their tolerance of your silly mistakes.
Thank you so much again for your time. And at the risk of
Go ahead.
at the risk, I'm going to try and pan over the audience so you can see who you've been talking to.
I'll hold that for you.
Thanks.
Thank you,
So-
Dr. Sharp. Everyone could wave and give him a.
Whoops.
Thank you very
Thank
much.
you. Thank you.
And Dr. Sharp, thank you so much for your time. We really appreciate it. This has just been so illuminating and really made this such a special community event. And I just wanted to thank you for for the generosity of your participation.
Thank you. I very much appreciate your interest and the questions from the audience. Thank you very much. Have a good evening.